video to give you a brief overview of some exam tips and tricks for your biology exams for both paper 1 and paper 2. So I'll start by giving some general advice first, before then going into sort of more specific bits of advice that pertain to each individual topic in order across both papers. So it will be quite a long video, but of course you can skip through it at 2 times speed, and obviously just go to the topics that perhaps are relevant to, for example, the upcoming paper 1. But as a little bit of general advice to start with, I think it's really important that you remain as calm as possible during the exam and in the sort of days leading up to it. If you stress, your body triggers the fight or flight response, and adrenaline, cortisol, etc. are released, and obviously have their effects. And one of those, as you know, is sort of increasing heart rate, etc., pupil dilation. But the biggest one, really, for me in terms of exams, is actually you get brain fog. And what that means is you tend to forget bits of information that your brain perceives as not particularly useful to your long-term survival or short-term survival. So your body's kind of gearing up for sort of immediate stress, like a saber-toothed tiger has just walked into the room and you need to either run or fight it. But of course there is no saber-toothed tiger, it's all in your head, but the response is the same, right? If you what you need to do is be able to fight the tiger, so you need to contract your muscles, etc. But if you end up sort of not doing that and instead remembering all the sort of formulae for area or volume of a sphere, you'll get your head bitten off pretty quickly. So the big thing is, and I know it sounds simple, but it's the biggest thing you can do before the exam, sort of in the day or two beforehand, is actually just to think, I've done all the work, I'll do as well as I can, but I'm not going to stress. And actually it's a difficult thing to do, but trust me, you will do better in the paper and you will think more clearly if you are not stressed. So as I mentioned there, I think it's important to have faith that the revision process that you've been through and the 2-3 years of study that you've done at the school is in there somewhere. And it's kind of your time to shine in a sense, right? It's to sort of almost show off what you do know, as opposed to worry about the things that you perhaps have forgotten. Importantly, I think there are certain types of question where you can gain time. So things like multiple choice questions are usually very quick to answer, and definitions are very quick. If you just know your definitions inside out, they're very often 2 marks, if not 3. And in the exam, of course, if you work to a mark a minute, you can puke up a definition in about 30 seconds, and that will save you perhaps 1.5 to 2 minutes, something like that per definition. Same with multiple choice. It should be a fairly quick bit of conceptual understanding that you just need to puke back up. And that will save you time in the long run to attack the sort of more mark-rich questions, things like data analysis, the Corms question, and just make sure you get all the marks on those. Write clearly, and you can use bullet points. So it's been confirmed by the exam boards that they like bullet points. You still need to write in good English and sort of using coherent structure, but you can absolutely bullet point your ideas. You do not need to write in full continuous prose. So long as what you write in each bullet point makes sense, I think that's a very useful way to attack questions, certainly the sort of 3 markers, 4 markers, etc. It's a very useful thing to do and to get into a good habit of doing, because it will help you organise your thoughts more clearly, and you're therefore more likely to get, first of all, get marks, but also not repeat information that you may have said already, and kind of get yourself muddled up in terms of the wording of your answers. If you were to look at mark schemes, and that kind of leads into this point, actually a lot of the marking points for these sort of 5 markers and things like that just have a collection of keywords. And I'll show you some examples later of that. But if you look at mark schemes, it's not how necessarily you say something. It's, have you been clear or said that, have you got around to saying that there's an idea here that there's more mitochondria and therefore more aerobic respiration, right? So as long as you've said that, it doesn't really matter how you say that, right? The mark scheme will just have mitochondria and aerobic respiration as the key marking points. So don't worry too much about how you're um, constructing your answers necessarily. Just go for the detail. Really important one, if you're confused by a question or it looks unfamiliar to you, you're clearly misinterpreting what the question is actually asking of you. So if you have read a question and you just think, you've never heard of this particular situation before and you're confused about what's going on, you're not actually interpreting the question in the right way. So you need to just calmly go back and think, right, I've clearly misunderstood something here about what it's getting me to do. So I'm going to reread the question and have in the back of my mind, what is it actually asking me? And more on that in a minute. Treat examiners like idiots. This is really important because, very often students, write exam answers and they'll write a fantastic answer, but they will leave out a bit of detail that in their head is obvious and is not even worth stating. And it really is important to state the obvious, even if you think it's so simple that it's not going to gain any marks. 9 times out of 10, it will get you a mark. Things like if they ask you a question about translation, about going from an mRNA sequence into an amino acid sequence, and they ask you to describe it, but they don't give you the word translation, tell them it's called translation. At the end of your answer or at the beginning, say, this process is called translation. If they're not telling you that, it might well be a mark. Always ask yourself 2 questions. What is the question really asking me, which we'll come back to, and I'll show you examples of that. And is there anything I've missed out, right? Is there any bit of small detail that I think or I'm assuming to be common knowledge that actually is worth putting down in an exam answer? And finally, don't repeat stuff in the question. The amount of times I see things where you're just puking stuff back up that they've already given you in the question, don't waste time. Okay. What I'm going to do now is go through some a little bit more sort of generic advice to sort of most exams before I then go into the detail of the actual syllabus. So this approach is something that I teach my students. I've sort of coined it as the backpedal approach. And I think this question sort of shows you where this can be used effectively. So explain how having more red blood cells is an advantage to athletes who take part in long distance races. And in my head, I'm highlighting the fact that they're asking me about having more red blood cells and how that is an advantage to an athlete who's clearly having to run in a long distance race. Very often, students will attack this question by starting at the beginning of the sort of rubric and try and work their way to a conclusion about how this means athletes can take part in long distance races. That is how you structure your answer, but it shouldn't be necessarily how you structure your thought process before you answer the question. So in my head, I think this is the perfect one to actually, sometimes in these types of questions where you're having to go from sort of A to B, actually start at the end, right? Start at the long distance race and the athlete. So in my head, I'm going to imagine an athlete taking part in a long distance race. What are they going to need? Okay, so I'm not going to look at the red blood cell component yet. I'm just going to, in my head, work out what they need, biologically speaking, in order to take part in the race. And of course, they're going to have to contract muscles. And they do that by using energy. Okay, so that's both of them here. So again, biology, right? Don't just say that they have to run, they have to contract muscles. Again, state the obvious. And they need energy to do that, okay? The energy, of course, the energy currency of the cell is ATP, adenosine triphosphate. And then I'm going to start saying, well, okay, how do we get ATP? Where does this athlete get ATP from? It is, of course, from respiration, both anaerobic and aerobic, of course, predominantly aerobic in this case, because we need a lot more oxygen. going to provide oxygen in the form of oxyhemoglobin, because, of course, don't forget we're talking about red blood cells. So I'm going to mention not only that they contain oxygen, but actually the oxygen is bound to hemoglobin within the red blood cells. Again, that links to a shell topic about how red blood cells are adapted to their function. Where do you get the oxygen from? Well, of course, this is to replace something called the oxygen debt. So when you're taking part and you're running a lot, you're doing a lot of this process. If you run out of oxygen, you of course start to respire anaerobically, produce lactic acid, and the amount of oxygen that you need to sort of break down the lactic acid and continue aerobic respiration is the oxygen debt. The final thing is to then say, well, if you've got oxygen because you've got red blood cells, that means if we have enough oxygen, I have less oxygen debt, and I can run further and for longer, which of course is important to my athlete over here. And of course, where does the red blood cell component come in? Well, it's the hemoglobin, right? So how is having more red blood cells an advantage here? Well, I'm going to be able to have more oxygen carrying capacity. I'm going to have more hemoglobin to carry more oxygen. If I have more oxygen, I can do more respiration. I can produce more ATP to contract more muscles and obviously run further without fatigue. So if you were to look at the mark scheme for this, again, it's in various bullet points as opposed to continuous prose. And we've got two aspects to this. This is essentially everything I've just said by doing my back pedal approach, starting at the end and working my way back to why we need more red blood cells. And you can see that the two sort of parts of the question here are answered. Why do we need the red blood cells? Well, it's for oxygen and hemoglobin. Why is this an advantage to athletes? It's because it will stop fatigue. It allows more energy to be released in the form of ATP. And that means the athlete can run faster or longer or further. And that's really important. You cannot just say, oh, it means that the athlete can do more races or can run more. What we mean is faster, longer, or further. So again, be specific. But I think this back pedal approach is really useful for these types of questions where you're just having to describe a process essentially. Sometimes handy to start at the end and kind of consider what you need for each step, working backwards to the start. So the next thing is to ask yourself what the question is truly asking you. So I've picked a couple of examples here. The first one is it shows you a sort of circulatory system here with a heart. It's a funny looking heart compared to what we're used to in humans. And this is, of course, it says the word gills up here, so we haven't got lungs, we've got gills. But this is the gas exchange organ, gas exchange system. So it's the equivalent of the lungs in humans. And we've got the body capillaries down here. So essentially it's similar. And we're asked to describe how the structure of the heart differs from a human heart. And what is the question really asking? Well, again, it's 3 marks, so it's worth doing this. It's asking about the structure, and I would highlight that in the exam. It is not asking about the difference between, the blood or anything else. It is literally the heart and the structural elements within it. So I would zoom in to the heart and think structurally. So you cannot talk about blood pressure or oxygen concentration or carbon dioxide concentration, because that is not a structure. Words in the question are important. It is saying, name some structures that you can see, some physical differences that you can see in this exam picture, and compare that to the human heart. So automatically, there's only one valve here, right? It looks to be that there's only one atrium and one ventricle. There's only 2 chambers, therefore. So it's automatically we can see some of these differences. So again, structure is really important. And if you look at the mark scheme, again, it's kind of as long as you get the idea that there's fewer valves or fewer chambers, you get the marks. But importantly for this, what is it asking? It's asking about structure, not type of blood or pressure or blood flow. So that is why here the mark scheme rejects anything to do with blood flow or the circulation itself, so pressure or what have you. Again, this is a similar type of question. Sometimes a baby has a hole in the septum here between chambers A and B. In this case, these are the ventricles. So chamber A is the left ventricle, chamber B is the right ventricle. Don't forget it's flipped. The other way I know that is because the heart has a lot of more thicker muscle here and more sort of tissue on the left side here, because we're going to have to pump with greater force, so that the contractions have to be more forced, and therefore you get a greater pressure of blood leaving the left ventricle through the aorta to the rest of the body. And of course the aorta is this branched thing up here. So if you have a hole between these two, what condition does this have on the baby? Now, you may be fooled into thinking this is a sort of off-spec question, but it really isn't, because what is the question truly asking you? It isn't asking you about a condition that the baby has, really. What it's saying is, if you have a hole between chambers A and B, what's the outcome? So basically the question is asking, do you know what chamber A and B are? And we do, it's the left and right ventricles. And do you therefore know what happens if the blood in each mixes with each other? Okay, so what the question is truly asking is, do you know what the blood looks like in chamber B compared to chamber A? And of course, chamber B, the right ventricle, it is deoxygenated. And of course, in chamber A, it is oxygenated. So that's what the question is really asking. Okay, so you can obviously say that the blood, it allows blood to mix, but really it's asking this, okay, which is that the oxygenated and deoxygenated blood mix, and therefore you get less oxygen on the left side, you get more deoxygenated blood in chamber A, and therefore more deoxygenated blood or less oxygenated blood then goes to the rest of the body for less respiration. Again, I'm going quickly through it because this isn't supposed to be a teaching session, but I hope that makes sense. Just one point, if you're talking about valves, the bicuspid and the tricuspid valves here, you actually do not, you're not required to know, for example, in this case, what structure C is in terms of it being called the tricuspid. You can literally just call both of them either the right or left AV valves, atrioventricular. They separate the atrium and the ventricle. So if you always seem to not be able to remember that, the right side of the heart is the tricuspid and the left is the bicuspid, then just call them both AV valves, and that's fine. Again, another question here is what happens to protein in the human stomach, the fish protein? And quite literally, this is saying what happens to protein in the stomach. So we're thinking hydrochloric acid and protease, so things like trypsin and pepsin. And that is hopefully what you can see here. Okay, so again, I'm not going through all the answers, but key thing is, what is it really asking? It doesn't matter that it's a fish, that it's fish protein. Doesn't really matter what protein it is. It's just saying what happens to protein in the stomach. On that note, another type of question, very similar, it's essentially the same question. What happens to meat, i.e. protein, when it is in the hawk's stomach? And you have probably not done the digestive system of a hawk in class, but you don't need to know that it's a hawk. Again, highlight meat or protein in this case, and stomach, right? What happens to protein in the stomach? And the mark scheme is essentially the same thing. You are not, boys may sort of get a bit confused that it says the hawk stomach. Don't worry about it. It's not important, right? The important bit of the question is asking you what happens to protein that you eat in your stomach. Okay, and you haven't done the hawk's digestive system, but you have done human. So again, important to know what the question's really asking. As I said, mark schemes are often in bullet points. So if you're explaining a reflex arc, if you were to look at the mark scheme for this, it's quite literally in bullet points, right? So long as you have everything in the right order, so long as you've mentioned receptors sending signals to sensory neurons, and then you've maybe mentioned the CNS or the coordinator or the synapse and then an effector, you get the marks. So it's not necessarily about how you construct your answer, or kind of, the sort of prose that you're using. It's, do you hit these key points? Okay, so again, really important. And that's why actually I would answer this question in bullet points. Okay, what I'm now going to do is go through every topic. So this is going to be a little bit of a marathon for me. But I'm going to go through every topic reasonably quickly, or as quickly as I can. I'm not going to reteach anything, but what I'm going to do is pick out the key points of each topic that I think are really important to make sure you know. Maybe it's things that the exam board would like to test you on, maybe it's key words or key phrases that you need to write for that particular topic in order to score marks. Okay, so I'm going to go fairly quickly. Obviously, if you've got any issues, you feel free to email me or speak to your biology beak. But these are just key points for you to make sure you know in each topic. So I'm going to start at the beginning. The first is is maths, which is how you work out percentage change. And just remember that we do change over original, so the difference from the start to the end value, divided by the original value, and we multiply that by 100 to work out percentage change. If you're asked for relative change or the change, you don't multiply by 100, but if you're asked for percentage change, you then have to multiply by 100. Either way, change over original. I've put the owl there because I teach my students to coo like an owl to do percentage change. So, variety of living organisms. The first thing here really is to remember that fungi demonstrate a type of nutrition that's called saprotrophic. So they are saprotrophs. And saprotrophic nutrition is a term you need to know. And of course, this means basically extracellular digestion. So these organisms secrete enzymes by secreting enzymes into the environment, onto the material they need to digest. They are digestive enzymes and they absorb the products. Okay, so it's called saprotrophic nutrition. Protists or Protoctista are defined by exclusion. Okay, so they are essentially defined as a eukaryotic cell, so they're single-celled, they're eukaryotes, but it is not a plant, animal, or fungus. Okay, so it's sort of like it's defined by exclusion. You need to know two particular types. So you need to know the Plasmodium species. These are responsible for causing malaria, so they're pathogenic. And trypanosomes are another example of a protist, again pathogenic and can cause African sleeping sickness. So worth bearing in mind. I think malaria is the key one, but you are required to know both of these. So Plasmodium are protists that cause malaria, trypanosomes, sleeping sickness. Remember that viruses are not alive. They do not do the sort of processes of Mrs. Gren or Mrs. Gren C. C, of course, standing for control or homeostasis. So you may have learned this as Mrs. H. Gren instead of Mrs. Gren C. But anyway, a pathogen, again, living organisms. I mentioned that earlier. These are both pathogens here, but bacteria, of course, are as well. Fungi are. And you need to know that the definition, by the way, I'm going to do definitions in red. So anything in red is a definition you need to know. Pathogen is a or any microorganism that causes disease. So typically, bacteria do this by damaging cells and producing toxins, and viruses, of course, invade cells and kill them directly by reproducing inside cells. You need to know the differences between excretion and egestion. Do not get these confused. And the same thing as well with respiration and ventilation. So students tend to get these all muddled up. Just remember, if you're ever going to, as we saw with the athlete, if you're going to talk about movement in any way, shape or form, whether that's running or weightlifting or what have you, that is because you are contracting muscles. So contraction of muscles is the key thing there. And of course, there are three main types of reproduction for cells to do. There's mitosis, meiosis, which is to form gametes. So mitosis is just for sort of normal repair of eukaryotic cells, typically. So just for growth and repair of tissues. Meiosis is specialized for to the germline, to the germ cells, so the ovaries and the testes that produce the egg cells or the sperm cells. So these produce haploid cells, but it's a specialized form of cell division. And bacteria do a different thing, similar to mitosis, but they essentially do a different thing. It's called binary fission. And again, look that up if you're not sure. I've put some definitions up here for you to read in your own time, and here as well. So again, these are the four things that students tend to get confused quite a lot. They will say egestion when they mean excretion and vice versa. And when they mean respiration, they often say ventilation. So do not get these mixed up. Especially these two come up quite often in terms of getting this correct. So, cell structure. Cell membranes occur in all cells. So don't forget that is one that you should always be labeling. The difference is, or one of the differences, of course, is when we come to the cell walls, which sits outside the cell membrane. Eukaryotes do not have cell walls. but animal cells or eukaryotic cells do not have cell walls, but plant cells and bacteria most certainly do, and of course fungi. Ribosomes are the site of protein synthesis, and the important point here is on diagrams they are smaller than mitochondria. So if you're not sure which one the ribosome is, it's the smaller one. Okay, they are much smaller than mitochondria. Mitochondria are the site of aerobic respiration. That's important. So it's a key adaptation of cells to meet high metabolic demand is to have mitochondria. This is for aerobic respiration, not anaerobic. Again, just to differentiate between plants and fungal cells. Plant cells, don't forget, you need to know they have chloroplasts. This is special to plant cells. They have chloroplasts and contain chlorophyll. So chlorophyll is the pigment within chloroplasts. This allows for photosynthesis to occur. The cell walls of plant cells are made up of a polysaccharide called cellulose. You need to know that. It's incredibly strong and allows turgor pressure to be maintained. It's very strong. The cell wall, the strength of this and the turgor pressure allows resistance to osmotic lysis, so resistance to water movement, and therefore swelling and puncturing of the cell, which happens in eukaryotes because they don't have a cell wall. So they will, if water moves in and enough of it moves into their cell, they will swell and burst. And plasmolysis, this is really important, causes shrinkage of the cell membrane to detach from the cell wall. But that is because water is moving out of the cytoplasm, and it's filling the space between the cytoplasm and the cell wall. It is not the fact that water is moving in from the outside. Okay, so it's coming from the inside of the cell, water moves out. And starch is the main storage polymer. For fungal cells, you've got something called chitin, which is again it's sort of like cellulose in a sense. It's a polymer in the cell walls, very strong. It's a reason why certain insects have chitin for exoskeletons. They use glycogen as their storage polymer, just like eukaryotic cells, just like human cells. And they carry out, as we said, extracellular digestion. So they're saprotrophic. Okay, and of course, fungi generally look a bit like this. I mean, this is a very simplistic version, but you have hyphae, which are these long chains of cells sort of stuck together, and the whole structure is called a mycelium. So looking at organelles, you need to know the definition, which is here. It's a membrane-bound structure that, within a cell that has a specific function. So classic examples, chloroplasts in plant cells, nucleus in all living cells, apart from bacteria, of course, they don't have a membrane-bound nucleus. Mitochondria and ribosomes. Okay, key thing when you're drawing or labeling, mitochondria look like this. So they're kind of they have this sort of folded inner membrane, but chloroplasts have a couple of membranes and they have discs here. So that's how you recognize a chloroplast as opposed to a mitochondrion. Important to remember that. A note on protists or protoctists. They look similar to eukaryotes. They are eukaryotic in nature. It's just that they are not a plant, an animal, or a fungal cell. So they're defined by exclusion. They are eukaryotic. But they have prokaryotic features. So you can get caught out by this, where they show you something like this, but because it's got a flagellum and it looks a bit like a bacterium, students always go, oh, well, this is a prokaryote. When actually what you look at is it's got a membrane-bound nucleus, it has mitochondria, so it's got organelles within it. It's got chloroplasts, in fact. So that's definitely odd, because that's typically what we find in a plant cell, which is definitely eukaryotic. So really important to be clear about that. And of course, the flagellum is very common for bacteria, but because it's got the sort of mitochondria, the chloroplasts, the membrane-bound nucleus, it's got bacterial features and eukaryotic features, and therefore it is probably a protist. They have features of both. Okay, so again, if you're confused whether it's plant, bacterium, or animal cell, it's probably a protist or a protoctist. Okay, it can have features of both. Magnification, you need to remember your unit conversions, which is this. So just remember you are expected to be able to interconvert between all these things. And it's important to do it in stages, right? Don't try and just be clever and jump from meters to nanometers, right? Take your meters value times that by 100, then times by 10, then times by 1,000, then times by 1,000 again, just to be clear that you are actually getting to nanometers and not skipping any stages by trying to merge by, going from centimeters here to micrometers. You would have to times by 10,000. Okay, but better to times by 10 first to get to millimeters, then times by 1,000. Okay. If you're working with cells, generally speaking, you'll work in micrometers with various drawings or microscopy calculation. 1 millimeter is equal to 1,000 micrometers. Remember that. That's kind of the key one. And remember this equation. Okay. And remember that the image here, so actual size is image over magnification, or magnification is image divided by the actual. Make sure your units are in the same. Make sure you're in the same units. And remember that the image is not real, right? The image is the picture on the paper, right? It's an image that's been printed. Okay, actual is the actual thing in real life. Okay, stem cells. So again, make sure you know your ethics between embryonic stem cells and adult stem cells. Know your adaptations of these particular types of specialized cell. So these have all been derived from stem cells. And you need to know that stem cells, of course, can differentiate to produce specialized cells. These are examples that you need to know in terms of their structure and being able to label them. Just a point, just to remember when you're saying about how red blood cells are adapted to their function, everyone talks about hemoglobin and being biconcave, but everyone forgets why being biconcave helps. Firstly, it increases the surface area to volume ratio, of course. But the other thing is it allows the red blood cells to be quite flexible and deformed. And that's really important when they are traveling through very narrow capillaries. They need to be quite sort of malleable. Okay, and the fact that you are biconcave in shape allows them to move through capillaries a lot easier and be more flexible. I think that's all I need to know there. Definitions, really important for transport in and out of cells. This topic, this is osmosis, diffusion, active transport. You need to know them because you will get one of them. I'm almost guaranteeing you will get one of those in your exam. Make sure when you're talking about adaptations of how certain molecules or compounds get into and out of cells, it boils down to this particular equation, which my divisions will be well aware of. And this is how you describe how certain features are adapted to transport things in and out of cells, because it always comes down to diffusion or active transport. Linked to the question. So, for example, more diffusion means more nutrients. Now, I hate the word nutrients, so use glucose or amino acids or something. Be clear about what you mean by nutrients, but link it to the question, right? So why does the deer need to do more diffusion and have a high surface area to volume ratio? Well, it needs to get more glucose, and that's needed for respiration to produce ATP to run away and survive, right? So again, just give all the detail as much as you can, right? Link it to the question, right? Why does it matter for the deer, right? Don't just say, oh yeah, to get more glucose for respiration. Well, why does the deer care about that, right? That is to get more ATP, to do more muscle contraction, which I've forgotten to put here, to run away from predators. Okay, so for the deer. Remember the potato osmosis practical. Key thing to remember is when you're dissolving anything in water, so ions, amino acids, glucose, whatever it is, that reduces water potential. Okay, this could be ions, of course, right? So if you're putting ions into solution, more ions move somewhere, you're going to reduce water potential. Okay, and that's just shown here. So your solute here might be ions, so I may be dissolving sodium ions or moving sodium ions across my membrane for whatever reason. And water molecules, which are shown here, attach to these molecules like these ions, for example. And you can see here that on both sides I've got the same number of water molecules, but here a lot of those water molecules have attached to the solute, almost like Velcro, and that means they are not free to cross the membrane. The reason is the ion here, or if this is glucose, whatever it is too big to pass through the selectively permeable or partially permeable membrane. But a lot of these water molecules can move that way. So you've got fewer water molecules, free water molecules here that can move this way, but most of these can move that way. So again, lower water potential here, higher water potential here, and therefore water will have a net movement from right to left in this case. These are your definitions. I highly advise you to remember these because you are almost guaranteed to get one of them in either paper 1 or paper 2, just to puke up for a couple of marks. Okay, moving on to the heart then and circulation. So again, this was another shell topic. Make sure you know your adaptations of blood vessels. Don't forget that valves open and close due to pressure changes on either side. So make it very clear, the blood pressure in the ventricles. So, ventricles contract, blood pressure in the ventricles exceeds that of the aorta, and therefore blood moves from the left ventricle into the aorta, and the semilunar valves open. So just be very clear about that. Don't forget to identify the left and the right sides. Key one is to look for the thicker wall on the left-hand side, but also the vena cava connects to the right atrium. So you can always identify the vena cava as well. Sometimes in the mark schemes, they allow you to say that the left side is always on the right when we look at it, and vice versa when it's drawn. But that's a pretty weak argument. Okay, always look for the thicker wall and the vena cava. Hole in the heart, make sure you can answer those questions and think about how your examiners might test you on that and word the question differently to basically ask the same question. The heart rate, just remember to name the particular region of the brain that deals with controlling the heart rate. So that's the medulla oblongata in the brainstem. And of course, this sends signals down either accelerator nerves to increase the frequency of heart contractions, or deaccelerator nerves to decrease it. Make sure you know your double single circulatory systems and what that means. Renal, of course, when you're looking about blood vessels, refers to kidney. Again, make sure you know what that means. Renal means kidney, so renal artery, renal vein, etc. Pulmonary means lungs. That's the posh word for lungs. So pulmonary artery, pulmonary vein has something to do with lungs, either going to or from the lungs. Hepatic means liver. So make sure you remember that one. And of course, the hepatic portal vein is a key one because that goes directly from the intestines to the liver before it goes to the heart, as opposed to the hepatic vein, which goes straight from the liver to the heart. Remember, the coronary and the carotid arteries branch from the aorta. So it's sort of that snaky shape at the top and it branches off. The coronary artery goes to the heart and the carotid arteries, of course, go to the brain. And I've put here, just remember, when you're talking about adaptations, if you're saying that there's more muscle in the heart, I don't know why that's pointing to blood vessel, that's supposed to be the heart, always say there's a thicker wall. Say both. Very often, mark schemes will have both. There's more muscle, therefore it's thicker, and therefore we contract with more force on the left-hand side. If a question asks about the composition of blood, again, read the question as opposed to anything else, then that's always talking about what it's made up of. So in this case, it's talking about gas exchange. Okay, it's talking about oxygen concentration and carbon dioxide. Say both. Okay, don't just say, oh, there's high oxygen concentration in, I don't know, the arteries. Say there's a high concentration of oxygen in the arteries, low in the veins, but in the carbon dioxide, we've got a low concentration of carbon dioxide in the arteries and much higher concentration in the veins. If we ask about a feature of blood, then that's talking about pressure. Okay, so composition is talking about what it's made up of. Feature of blood is generally going to be referring to pressure. Okay, and of course, the left-hand side of the heart, arteries have much higher pressure than veins. Okay, gas exchange in humans. Make sure you remember the mechanism of breathing, and if in doubt, just say the intercostal muscles contract. So whenever you're going to talk about those, you don't need to refer to external or internal intercostal muscles, which are certainly in my notes. You can just say intercostal muscles contract, and you'll be right either time. So don't forget, talk about the sort of intercostal muscles first. So in both cases, just say they contract. Then talk about the thorax. Okay, this is the space sort of outside the lungs in the chest cavity. And of course, everything is down to pressure. So air moves in certain directions because of pressure changes. And we need to talk about the diaphragm. I'm going to talk more about that in a minute, but you need to make sure that you talk about all those things when you talk about the mechanism of breathing. So what's happening to the intercostal muscles, what's happening to the thorax, what's happening to the diaphragm, what's happening to pressure. You need to know, as you look down the wall of the bronchus, what these structures are. So you need to know that the ciliated cells here are called ciliated epithelial cells, and they have cilia to kind of waft stuff and mucus towards the mouth. And the goblet cells here, which look a little bit funky, they don't have these cilia on top. These are goblet cells, cell A, which produces mucus. So the mucus is, of course, to trap dust and bacteria and things, and the cilia on the ciliated epithelial cells are sort of wafting towards the mouth, helping to clear any of that dust that's been trapped. So when you're talking about gas exchange, it all comes down to pressure. So this is a little bit of a teaching thing. Imagine we've got the same number of molecules in a given space. You're at much higher pressure if the volume of the container, i.e. the thorax, is smaller. Okay, so if you're in a smaller box, you can see that the number of particles per unit space, i.e. pressure, is much higher. In this case, this would be air part, so oxygen and all that sort of stuff. Here, I've increased the size of the box, and therefore I've lowered the pressure. Okay, there's more space, there's more volume for the same number of particles to exist in. And this box is kind of representing two things. The box itself, the space, is representing the diaphragm. The actual box itself is representing the diaphragm, so the thing that I can sort of make sort of bigger and smaller, I guess. And the bit in the middle, the actual space, is the thorax. Okay, so that is the space between the lungs, space outside the lungs in the chest cavity. Okay, so more on that in a minute. Key thing is to remember that air moves from high to low pressure. Really important. Okay, so just on this, air would move from this container. If I'd connected them together, air will move from this container into this container until there's an equilibrium maintained. So what I'm going to do is on these humans, I'm going to put a diaphragm and a thorax. But key thing is when the diaphragm contracts, we increase the volume of the thorax and vice versa. So what I mean by that is if we contract our diaphragm, I'm going to increase the volume of the thorax. I'm going to make this box, the sort of lines, increase in size because they're going to contract. And the space, therefore, the sort of grey bit, is going to increase. Okay? The alternative is true, which is if the diaphragm relaxes, we then make it smaller, right? The thorax decreases, the volume, the grey bit decreases, and therefore, of course, in the top one here, the pressure is lower, and down at the bottom, the pressure is higher. So I've represented that by the letter P. So big P for high pressure, low pressure here. Sometimes it's difficult to sort of picture that though. So I've essentially put the diaphragm in red here in these two people. The thorax, which is the volume of the chest cavity that the lungs occupy, is shown by the white space, right, which is the sort of black bit, black box. Okay. So when the diaphragm contracts here, it actually goes from its normal shape, which is dome-shaped, to this shape. Okay, so its normal shape is like a fist, and you contract your muscles to flatten your hand out, and that's what contraction does. Okay, so if you contract the diaphragm, you can see here the volume of the thorax, the volume of this box has increased. Again, here we've created this picture where my pressure in here, the pressure inside the thorax, decreases. I've got lower pressure here, equivalent to this box up here. If the diaphragm relaxes, the thorax goes back. The diaphragm goes back to its original position, its resting position, which is sort of dome-shaped. The thorax has less volume, so the volume of the thorax decreases. I still have the same number of air particles in there, but they're going to be at a much higher pressure. So I end up with this situation here. And hopefully what you realise is where, if I put these P's on here, so again, I've got high pressure here when the diaphragm is relaxed, because I've got a decreased volume in the thorax. When the diaphragm contracts, I've got lower pressure because the volume of the thorax increases. And that's going to affect, of course, the direction of air movement, right? Air moves from high to low pressure. So if I've increased the pressure in my thorax, the air pressure in the thorax is going to be higher than the atmosphere. Therefore, the air particles here get forced out. So that's expiration, breathing out. And on the right-hand side here, I've increased the volume of the thorax. The pressure's decreased in here. So the outside atmospheric pressure must be higher, and therefore air will be drawn into the lungs. Again, all down pressure gradients, right, from high to low pressure. Really important. Again, we can use the backpedal technique to kind of describe what happens. So imagine the question's asking you about how you breathe out, what's the mechanism. Well, to talk about this, it's very nice, I think, and I still do this, to start here, right? To think, okay, air pressure. If I need to breathe out, I need to do this, right? I need air to move from my thorax out to the atmosphere. So if I need that to happen, I need a higher pressure of air in my thorax and a lower pressure outside. I can't control the air pressure outside in the atmosphere, but I can control the pressure in my thorax. So how I get air to move out is I need to increase pressure in my thorax. I do that by decreasing volume. Okay, so here, decreasing the volume of the thorax. I therefore need the diaphragm to relax and become dome-shaped. And then that will allow higher pressure in the thorax, lower pressure outside. Okay, so that's what I'm showing here. So I would start with pressure. Pressure in the thorax needs to be higher. To generate this, the volume of the thorax needs to decrease. This is done by relaxing the diaphragm, allowing it to become dome-shaped, and the air moves from high pressure in the lungs to lower pressure in the atmosphere. Bingo. But there's one thing we haven't talked about, which is the rib cage. And this is very easy. Don't work, don't sit down and revise this. Just do this in the exam. Doc H tip is what happens to the ribs. Put your hands on your rib cage, breathe out, and you will feel your rib cage move down and in. Okay? If you, of course, breathe in, what happens? You can feel your rib cage expanding, right? You can feel your rib cage moving up instead of down and out instead of in. So always remember to say that, okay? This is as a result of intercostal muscles contracting. So always mention intercostal muscles, and we just say they contract. Doesn't matter whether you're breathing out or in, okay? What changes is how you word this and how you word this, okay? So if I was going to breathe in, I would flip this around. Rib cage has to move up and out. The intercostal muscles contract, and I would then go through this entire process, but obviously vice versa. So pressure in the thorax needs to be lower than the atmosphere. The volume of the thorax must increase. This is done by contracting the diaphragm for it to become flat, etc. Really important, okay? So if you forget what happens about the rib cage, don't forget the rib cage, don't leave it out. Put your hands on your rib cage and decide whether your ribs are moving down and in or up and out. And this is always to do with the intercostal muscles, really important. Okay, food production is the next topic. Just remember, in terms of what organisms you use, if you're making bread and beer, hopefully you may have done this before, you add yeast. You don't add bacteria, okay, or protists, right? You add yeast, right? Fungi, okay? Don't forget yeast are a type of fungus. Yeast respire anaerobically to produce your carbon dioxide and ethanol. So in the case of bread, I really like the carbon dioxide to make the bread rise. In the case of beer and wine and things, I want the ethanol. And this, of course, helps you remember the equation for anaerobic respiration in yeast, right? Glucose is immediately broken down in a lack of oxygen to produce carbon dioxide and ethanol. The other organisms you come across, so bacteria and humans, must do the opposite, which is produce lactic acid. Okay? And of course, in yogurt making, don't forget, you need to know the exact type of bacteria that we use. It's called lactobacillus. When it's doing respiration, it does not use glucose preferentially. It preferentially uses lactose. So really important. Okay, so when you're talking about making yogurt, we're going to talk about lactic acid being produced, but we're going to say that lactobacillus bacteria do this. They use lactose instead of glucose for respiration to produce lactic acid. This is going to, of course, lower the pH. It's acidic, and it's the lowering of the pH, which is why milk goes sour. And eventually, of course, the proteins in milk start to clump together and thicken into yogurt. And that's why you need to keep it cool at that point and not warm, because it will eventually go off, because you'll produce so much lactic acid and so much bacteria that it then goes off. Okay, so you then want to keep it at a sort of steady temperature, about 4 degrees in a fridge. Remember, of course, in terms of your gases, so this is gas exchange and gases in the atmosphere, remember your numbers. I think everything's pretty easy to remember. So nitrogen is exactly the same. Oxygen is 21 and 17. But people tend to forget the carbon dioxide one, whether it's 0.044. How I do this is I just remember that carbon dioxide, it begins with carbon, which makes 4 bonds, and therefore it must have the number 4 in it. And it's either going to be 4 or 0.04. Which way round is it? Well, use your brain to work out logically, right? When we inhale air, we probably don't have much carbon dioxide because it's mostly oxygen and nitrogen. But of course, your cells will then use the oxygen to respire, produce carbon dioxide, and you then exhale that. Okay, so carbon dioxide must have a higher concentration in exhale there, and of course that therefore must be 4, not 0.04. Okay. The other point, just to make sure that you're aware of, is humidity here is akin to water vapor. That's what it means, right? The concentration of water, gaseous water molecules in the atmosphere. Okay. So make sure you know that table. Final shell topic was biological molecules. Remember that starch and glycogen are your storage polysaccharides for storing glucose into big chains. Starch is in plants, and of course glycogen is in fungi and humans. Amylase is the enzyme that breaks down starch, plant sort of storage carbohydrate, into maltose. Okay? And remember, amapitator was how I taught Mylot. Remember that. Okay? Glucose and glucose makes maltose. So in terms of your disaccharides, your how you can build glucose up into larger molecules, 2 glucose molecules together make maltose. This is broken down by maltase, so you can go back from maltose to glucose using maltase. I remember that because Maltesers are the most boring chocolate in a pack of Revels. Apologies to all the Maltesers haters and lovers out there. I quite frankly think they're really boring. But therefore the most boring disaccharide you can possibly make is maltose, because Malteser, and therefore it must just be 2 glucose molecules. The next one is to swap one of the glucose for fructose, and that makes a disaccharide called sucrose, and this is pretty sweet. So this is table sugar, and of course it's digested. It's broken down into glucose and fructose by sucrase, so that's the enzyme. And I remember this because fructose, I know, is one of the sweetest monosaccharides out there, so therefore that's going to create our table sugar that we use in tea and coffee, etc. And I can swap it out for galactose instead, so glucose and galactose. That makes lactose, which is the sugar that I just talked about, where lactobacillus likes to use this sugar for respiration, and that's broken down by lactase. Okay, so you need to know those enzymes and these disaccharides. Okay, these complex, slightly more complex sugars than just the standard individual monosaccharides. How you remember that glucose and galactose makes lactose is quite literally the word galactose. The word lactose is in the name. Okay, so that's a nice little way to remember it. You need to remember your food tests. So in the case of Benedict's, remember to be descriptive, right? Don't just say that it's a light blue solution that turns red. Say that it produces a red precipitate. Okay, it's red because there's a precipitate forming. You could centrifuge it out. Lipids, don't forget you add ethanol then water, and you get a cloudy emulsion forming if there's a lipid there. Don't forget E comes before W in the alphabet, so if you ever forget which one to add first to your lipid sample, you need to add ethanol first, then water. Starch, again, you get this sort of yellowy iodine solution, the yellowy orangey iodine solution that turns blue-black in the presence of starch. And the Biuret solution tests for protein, okay? And of course, you start with light blue and you end up purple, okay? How you test for starch, you need to know this. Just a couple of points here. Why you do these steps is really important for you to know. You boil the leaf for 20 seconds in water. This will denature enzymes to stop any other chemical reactions happening, so any more photosynthesis, for example, which might affect your results. And it therefore stops confusing results, okay? So, you want to stop any more chemical reactions happening that may produce starch. So really important to do that. The other thing, of course, is to break apart the cell walls, so to release the starch, so that when you add the iodine solution later, that iodine solution actually has access to your starch. Okay, it's not contained within sort of starch vesicles within your cell. Okay, so it's to stop chemical reactions, but also burst the cell open, burst the cell wall. Why you add ethanol, you then boil it in ethanol or in hot water. It's to remove chlorophyll. Okay, it's to remove any chlorophyll there, which basically allows you to see the color change a lot more easily. Okay, and again, if you're describing how to carry out this, always say what you do and what the result will be, but don't forget to talk about safety goggles, use forceps, use a Bunsen burner, etc. Right, moving into the remove, we've got digestion. Again, make sure you know your definition for digestion. Physical digestion and chemical digestion both happen. Don't forget the enzyme you need to remember in the stomach is called pepsin, and of course you've got acid hydrolysis that happens as well. Digestion occurs up to and including the duodenum. After this point, after the duodenum, the first part of the small intestine, there is no more digestion that occurs. It is purely adapted for absorption. So the ileum, there's no digestion that happens there. It is all about absorbing your digested products. And because you're having to absorb stuff in the ileum, again, movement of molecules into or out of cells, we're wanting to get them from the lumen of the gut into the capillaries that are flanking the ileum, and therefore we need diffusion. So it's that magic equation that I showed you earlier. So surface area, concentration gradient, diffusion pathway. Make sure that you remember that bile emulsifies fats and neutralizes hydrochloric acid. And the key reason for this is we need to emulsify fats to increase the surface area for lipase to act, and we need to neutralize the acid because, of course, your contents have come from the stomach, and things like lipase and proteases and other enzymes that are now going to be used in the duodenum probably don't have an optimum pH of about 2, unlike pepsin, and therefore we need to make sure they don't denature. Key thing is to remember that bile is made in the liver, but it's stored and released from the gallbladder. So made in the liver, but it's stored in the gallbladder and released. And don't forget, of course, it's got both an exocrine function, the pancreas, where it releases the amylase, so pancreatic amylase, lipase, maltase, proteases, and of course the endocrine pancreas will come across in the endocrine system. Enzymes, again, key terms, right? Make sure you talk about your active site and your substrate. When they bind together, it's because they have complementary shapes, so you have complementary binding, and we then create an enzyme substrate complex. Always worth noting that word down. Make sure you know how temperature and pH affect the shape of the active site, and therefore this will link back to the enzyme substrate complex. If you change the temperature or the pH enough, it will change the shape of the active site because the bonds that hold it together will break, will change. That means that the active site is no longer complementary to the substrate, and we don't form enzyme substrate complexes, and therefore you don't form product. You need to know that enzymes are biological catalysts. This is all part of a definition. So you need to know why they're biological and why they're a catalyst. So make sure you get both of these down in an exam answer if you're asked what a biological catalyst or an enzyme is. Coordination and response, make sure you know what your neurons look like, so sensory, motor, and relay. Make sure you know what they connect to and from. Remember the way you remember about how your coordination and response occurs, right? You have a stimulus, a receptor, a coordinator, an effector, and a response. So silly removes, can't even read. The reflex arc, you need to know the features of this. So it's involuntary, it's fast. Only 3 neurons are ever involved, right? It has a single sensory relay and a motor, and of course very few synapses. So in that case, 2 synapses until you get to your effector. Remember for the eye to use my tips and tricks. So to work out what happens with accommodation, hold a pencil up close to your eyes and focus on something far away, and very quickly move your eyes to focus on the tip of the pencil, and you will feel that your eyes actually hurt, they actually ache, okay? And therefore something must be contracting, right? You can actually feel the ciliary muscles contracting, okay? So therefore, if you are focusing on something close to you, the ciliary muscle must therefore contract, okay? And then, of course, you need to talk about the lens shape. Don't forget, of course, to then talk about refraction of light. If you've got a thicker lens, you'll have more refraction, as opposed to when you have a thinner lens. And the whole point of this is to ensure that the focal point where the light rays converge is as close to the retina or on the retina as possible. If you're talking about pupil diameter, this is the James Bond thing. So imagine the James Bond intro of that kind of tunnel vision type thing, where this is your circular muscle and the radial muscle. They do opposite functions, they're antagonistic. Okay? So again, make make sure you're aware of these two. And if you forget the accommodation thing, use my trick. Endocrine versus exocrine, remember, so things like insulin, so the endocrine system, hormones. Insulin is a good hormone that you need to know. It lowers blood glucose levels. The endocrine system is essentially just using hormones. Okay, so it's defined as this. Okay, so the hormone is defined as a chemical secreted by a gland into the bloodstream directly that has an effect on a specific target cell by binding to a specific receptor. The exocrine system is actually just using enzymes and ducts. Okay, so anything that secretes an enzyme into a duct, into a tube, like the digestive system. So, the pancreas secreting amylase and lipase, that's part of the exocrine system. But of course, the pancreas also secretes insulin. So that's part of the endocrine system as well. So the pancreas actually does both of these. Plant nutrition. Photosynthesis, remember, does not need ATP. We do not include photosynthesis equation. We don't include ATP, rather, in photosynthesis equations. It is there technically, but it gets produced and then used up. So don't ignore ATP. That's not why we do photosynthesis. Just remember, it's an enzyme-controlled process, like respiration, like everything. So think about, if you're increasing the temperature too much, etc. If enzymes start to denature, if you change the pH, then potentially photosynthesis will be affected if you're a plant. You need to know what these three mineral ions are used for, so magnesium, nitrates, and phosphates. And again, I always remember what phosphates do. People forget this. The word ATP, the energy currency of the cell, stands for adenosine triphosphate. So no prizes for guessing what inorganic ion is used to make ATP, and the answer is phosphate. Okay. Of course, links to fertilizers and the fertilization topic. Not fertilization topic. It links to fertilizers. So things like phosphates, nitrates are very often used in fertilizers. You need to know your pondweed bubbling experiment. Talk about limiting factors. So you need to make sure that you think about which of these three factors are potentially limiting the rate of photosynthesis at any one time. You need to know that the xylem transports water and dissolved mineral ions, both of them, but only in one direction, from the roots to the shoots. Whereas the phloem transports your sucrose and your amino acids, so your sugars and your amino acids, in both directions. In terms of feeding relationships, make sure you know that producers are called autotrophs because, and these can be plants or bacteria. Autotrophic organisms, we've come across saprotrophs already, which use extracellular digestion to get their nutrients. Autotrophs don't need to do that. They are automatically doing this. So that means they can create their own food source. Basically, they can carry out photosynthesis to produce glucose. So typically plants, but some bacteria are photosynthetic as well. Remember that there's about 10% energy loss as you go up the food chain through trophic levels. So the reasons for this are generally here. You might be asked why. When we eat a lettuce, for example, why most of the energy that's in that lettuce, why 10% of it doesn't end up in us. That's because of respiration loss. So, potentially as heat. Movement, right? We have to move. We have to use some of the energy for that, and so does the lettuce. Or actually, I guess the lettuce wouldn't move, but if that was like a rabbit or something and we're having rabbit stew, then the rabbit would move. Not all of the lettuce that we eat will be digested, so we might have undigested bits of food. I've picked lettuce as an example, but it might not be the best example here, But certainly, bits of cellulose or like plant matter that we can't digest, sweet corn, for example. There'll be energy within that just then gets egested. Not all the food is eaten, so I might not eat all the lettuce or all the rabbits, right? So I might not eat all of it. Of course, you've got bones and things like that we don't eat, so not all of the food is actually eaten. And of course, one of the big ones, we excrete quite a lot of stuff. So in our urine and our etc. Breathing out, we breathe out carbon dioxide, right? We excrete water as well, water vapor and carbon dioxide. So energy loss occurs throughout all of these. You need to know the definition of a trophic level, which is here, which is the position of an organism within a food chain or web. Again, you could be asked that. In terms of reproduction, so for plants, pollination, you need to know the definition for that. And again, from anther to stigma, puke this up as written. Do not forget that, right? It's not just the transfer of pollen from a plant to plant, it's transfer of pollen from an anther of one to stigma of the other. Of course, it can be self-pollinated as well. You need to know your adaptations of wind and insect-pollinated plants. In the case of humans, you need to know that sperm is made in the testes but stored in the epididymis. So this is the sort of storage site for sperm, where they can kind of mature, obviously prior to ejaculation. This is kind of similar to the liver and bile, really. So remember, liver produces bile, and the gallbladder stores it. The analogous situation is here. So sperm is made in the testes, but it's actually stored in the epididymis. You can use two terms for the sperm duct, which carries sperm from the epididymis outside and obviously into the vagina in terms of sexual reproduction. So that's called the sperm duct, or if you want to be posh, and certainly in my notes it may be referred to as this, it's called the vas deferens. Sperm duct is fine. Fertilization occurs in the fallopian tube. Remember that, it doesn't occur in the uterus or the vagina. It occurs in the fallopian tube. And of course, the definition of fertilization is, in the case of humans, the fusion of a sperm and an egg nucleus to produce a zygote. It's the nuclear fusion, okay, to produce a zygote. Trophoblastic cells, when we start to differentiate, of course, you end up with the trophoblast on the outside and the inner cell mass on the inside of your developing embryo. The trophoblast on the outside, these are specialized cells that form the placenta, and the inner cell mass forms the embryo. And it's the cells of the inner cell mass that are embryonic stem cells. Okay. And of course, the placenta is adapted for diffusion. So remember the equation, we have to talk about surface area, concentration gradient, length of diffusion pathway, same thing as I described earlier. And a key point here for me is if you're going to talk about how concentration gradients are maintained high, always just say there's a good blood supply. That's pretty much always the answer, which is that there's lots of capillaries, lots of blood supply all around to maintain a high concentration gradient. Okay, moving on to inheritance in terms of genetics. Again, know your definitions, right? You need to know the definition of all of these things: gene, allele, locus, genotype, phenotype, your zygous state, so homozygous, heterozygous, dominant, recessive, and codominant. You all need to know the definitions. Okay, I will eat my hat if you do not get asked to define one of these in your exam. Remember, when you carry out genetic crosses, that if you're asked for the probability that the offspring have 2 different or 3 different phenotypes, so what's the probability that they will be a tongue roller and male? If you are asked for having a disease and being male, what's the probability of both things happening at the same time? You have to multiply the relative probabilities together. Okay, so use your genetic cross for both. Again, whether you're male or female is very easy. It's 50%, so you need to multiply by 0.5. And of course, work out the probability of having a disease, which depends whether it's recessive or dominantly inherited. But multiply together if you're asked for multiple outcomes in one cross. Always label your genetic crosses, label the outcome, so highlight the individuals, big B, little b, or little b. Highlight whichever one is going to be, I don't know, red hair. Highlight the ones that are going to be blue hair or whatever it is. Remember the difference here between DNA and RNA polymerase. Basically, when you're talking about transcription, you talk about RNA polymerase. For some reason, people have started to use the word DNA polymerase. That's not taught at GCSE, and it's not involved in protein synthesis. Okay, so we're making RNA, so it's RNA polymerase that does transcription. Okay, so forget DNA polymerase. RNA polymerase is the one. Remember, of course, that to develop new features as a result of mutation and kind of selective breeding and being able natural selection, it all occurs because of the same process, which is you have random mutation during the protein synthesis stage. That leads to variation in the offspring, whatever that variation may be. So it might be that they're able to degrade an antibiotic where their parents couldn't. That means they have increased ability to survive, and therefore they can reproduce faster and more efficiently, and they can pass on the alleles that they have obtained by mutation here, the variant alleles that are useful. They can pass this to the next generation. So you then get even more daughter cells being antibiotic resistant or whatever that may be. Okay, Don't skip out the step, right? They pass on the alleles to the next generation. Okay, important. Right, I realise I'm going quite quick, but that is kind of deliberate because, again, you should have done the revision. This is just sort of picking out some key points to remember. Final thing I just want to talk about is inheritance, okay? Which is hopefully you're all very good at doing the kind of genetic cross thing, standard genetic cross, right? So in this case, I've got two heterozygous individuals. They've got, big A and little a in terms of their alleles. So remember, alleles are each individual letter. The genotype is collectively what letters they are. So big A is a genotype because they've got two big A alleles. We've got alleles for red hair and blue hair. And in this case, I'm assuming that the allele for red hair is dominant over the allele for blue hair because it's a capital letter. And we can do the genetic cross like this. And I would highlight these three because we've got one copy of the dominant allele. Therefore, we will express this phenotype. So they will be red hair individuals. And of course, the probability of having a blue hair individual is only a quarter if I've got two heterozygous parents. But if I had a child and she looked like this, she's got red hair, and I know that the parents are both heterozygous, the question sometimes is asked, what's the genotype of this individual? And the answer is not easy because you do not know whether she is big A or big A little a. She could be either of these. All we know is she's not little a. And you may get a question that says, well, what's her genotype? How would you work it out? And this is where we do something called a test cross. Okay, and I'll show you what I mean here. So what I'm going to do is I'm going to take her, and she is either going to be big A or big A little a. And I don't know which one. Okay, so I'm going to take her genotype, and she's one or the other. She's either going to be big A or big A little a. I don't know which. What I'm going to do is take a test, and this is why it's called a test cross. I'm going to take a test individual. I'm going to take a homozygous recessive individual. And that's always the case. Okay, I take, in this case, an individual with blue hair. Okay, I'm going to get her to have a child. I mean, this could be done in rats or whatever. It doesn't have to be humans, but perhaps it's unethical to do in humans because you're forcibly mating. But anyway, you take this organism where we don't know the genotype, and you mate them with a homozygous recessive where I do know their genotype. It has to be homozygous recessive in this case because I would have to pick a phenotype, and in this case, I'd pick someone with blue hair. Okay. If I picked another person with red hair, I've got no idea what genotype they are, which is the same problem. So I pick an individual that I know from their phenotype what their genotype is. In this case, it's little a. So it's a homozygous recessive individual. And I look at the potential outcomes. And I've got two, depending on whether she, this individual here that I don't know her genotype, she could either be big A or big A little a. So we can do this, right, in theory. If she's big A, these are the options. In each case, the children will only ever be heterozygous. They will only ever get a big A from mum and a little a from obviously it would have to be a male in this case, homozygous recessive male. All of the options would only ever be heterozygous. In other words, all of these children would have red hair. The alternative is if this individual here, this lady, is heterozygous, so big A, little a, so one of these two, the outcome is different, right? The male would still be little a because that's the only alleles that he can give, but the option is different, right? It's a 50-50 ratio. Half the offspring will be homozygous recessive. Half the offspring will have blue hair, okay? So, how do you know? Well, you then look at the outcome of the crosses, right? And you cross this lady with this man here, and you look at the offspring. You obviously need to have enough offspring to count, and you go, okay, are all the offspring always red hair, or do we, across a few studies, do we get some children that have blue hair? And if the blue hair arises at any point, we know she must be heterozygous. If the children are always consistently red hair, in every mate that we do, every cross that we do, she must be homozygous dominant. She must be big A. Now, of course, you wouldn't really do that with humans. It's unethical. But if this was rats or horses or animals that you can quickly sort of set up crosses in a lab with sort of less ethical issues to consider, then this is absolutely a very sound way of teasing out whether she is big A, or big A, little a, is to look at the children that they produce. Okay, so again, this is what I've said here. If she's heterozygous, we would expect 50% of her offspring to have blue hair. If she's homozygous, children would only ever have red hair. Okay, homozygous dominant, of course, we mean. Okay, that's it. Best of luck for your exams. Obviously, do get in touch with anything, but hopefully that's useful. Not as a teaching tool, but as a sort of way to revise the key bits of information that I think for each topic it's worth remembering. So yeah, best of luck. Write in detail and don't worry about it. Don't stress. You'll do well if you approach it with confidence and clarity. So best of luck.